the classification of Oedogonium. Oedogonium belongs to the class Chlorophyceae, order Odogoneales, family Odogonaceae. The family comprises about 400 species and 3 genera. The 3 genera including in this species are Odocladium, Bulbocate and Odogonium. The Odogonium only possesses 285 species, that is why it is the type of the family. Now we will take the habitat of the genus Oedogonium. The habitat of the Oedogonium is freshwater. It usually prefers quiet conditions, quiet waters and avoid flowing waters. It is attached with the substratum by means of a rhizoidal cell known as holdfast. It also grows epiphytically on larger green algae leaves, petioles, stems of the aquatic angiosperms. It is also found in some quiet situations of ponds, lakes and streams. The thallus structure of Oedogonium is filamentous thread-like. The cells in the filament are placed end to end and the rhizoidal cell is fixed with the substratum. The free end of the apical cell is somewhat rounded and extended. The cell structure of Oedogonium is like this. The cell wall is conspicuously or inconspicuously made of 3 layers. The innermost is cellulose layer, middle pectic layer and outer chitinous layer. The outer chitinous layer prevents the loss of the pectic layer, so the filament looks like a wet thread. Just inside the cell wall there is cell membrane and the protoplasm is constituted of a large central vacuole which possesses the cell sap. The protoplasm also contains a large reticulate chloroplast in which are embedded a large number of pyrenoids. A parietal nucleus is also present in the cell. Certain cells in the filament possess ring-like markings of cellulose which are 1 or 2 in number. These cellulose markings are known as caps and the cell containing these caps are known as cap cells. The growth usually occurs simply by cell division. Usually in Oedogonium, certain cells in the filament which possess caps or cap cells, they undergo cell division. Not all the cells in the filament undergo cell division. It is only that those cells which possess the caps or called cap cells which undergo cell division. The cell division occurs here in 3 steps. First is ring formation, next is nuclear division and third is formation of new cells. The ring formation occurs simply by the formation of a scar-like structure just at the upper end of the cell. This scar gradually thickens and forms a ring-like structure. This is of a hemicellulose in nature. The nuclear division is this ring-like formation is followed by the nuclear division. The nuclear division starts by the movement of nucleus towards the upper end of the cell where it divides into 2 daughter nuclei. The nuclear division is followed by the formation of cytoplasmic strand between the two nuclei. The formation of two cells occurs by the breakdown of the outer two layers outside the groove, just outside the ring. And this breaking of the two outer layers permits the ring to stretch out. Now the cell is twice as long as the normal cell, and finally the cytoplasmic strand moves upwards just at the base of the groove, and finally the cell gets divided into two daughter cells. In case of Oidogonium, the successive divisions takes place only in the cap cells, so the number of caps formed on the cap cells determines the number of cell divisions cap cell has undergone. Now we will discuss about the reproduction of Oidogonium. Oidogonium reproduces usually asexually by fragmentation and spirulation, and sexually by advanced oogamy. The fragmentation in case of Oidogonium takes place either by dying off some of the cells of the filament or by accidental breaking. The fragments so formed by growth and cell division develop in the new plant bodies. In asexual reproduction, the zoospore formation is most common form, where certain cells of the filament, usually the cap cells, they develop into zoosporangium. Each zoosporangium produces only one zoospore. At the formation of zoospore, usually the cytoplasm recedes from the cell wall and gets collected in the middle of the cell. One portion of this zoosporangium, one portion of the cytoplasm present in the zoospore recedes and becomes hyaline, colorless. At the base of this colorless beak, a ring of flagella is formed. This ring of flagella are produced from the blephroplasts. So these blephroplasts are arranged in a ring-like structure. These blephroplasts are connected by a fibrous filament, so forming a circular ring. So from this blephroplast ring arises a single flagella from each blephroplast. So a ring of flagella is formed around the zoospore at the upper end, just below the beak. These zoospores are liberated out from the zoosporangium by the transverse rupture of the zoosporangium. On liberation, they move into the water for some time. After their movement, they settle down on the solid substances where they germinate. Before germination, they attach themselves with the substratum by this colorless beak, and this colorless beak gets converted in the rhizoidal cell that is also known as holdfast. The remaining part of the zoosporangium divides transversely and develops into the new filament of the thallus. This zoospore formation usually takes place during the favorable environmental conditions. Now we will take the acinids. The next type of spore which is formed in the Oidogonium filament during unfavorable conditions are called acinids. These acinids are thick-walled structures and are produced singly in some of the specialized cap cells of the filament. These acinids on maturity also are released in the same manner as the zoospores are released from the filament and develop into the new organisms on the approach of new favorable conditions. In Oidogonium, it is distinctly advanced type of oogamy. The sexual reproduction is, in case of Oidogonium, is by the formation of special reproductive organs, that is antheridia and oogonia. these antheridia and oogonia are developed by the special cells of the filament. So not all the cells of the filament produce antheridia and oogonia, but certain specialized cells having usually one or more cap cells at their apical ends which develop into the gametangia. Sexual reproduction in case of oedogonium is determined by certain external factors such as sunlight, concentration of carbon dioxide, and the deficiency of nitrogen. Depending upon the distribution of sex organs, the oedogonium has been divided into two species, macandrous species and nonandrous species. The two species of oedogonium can be differentiated from each other by the possession of small dwarf male filaments in case of nonandrous species, whereas these dwarf male filaments are not found in macandrous species. In macandrous species, usually the antheridia are produced on the filament of the normal size, whereas in case of nonandrous species, the antheridia are produced on small dwarf male filaments. The macandrous species may be monoecious or dioecious. In case of monoecious species, both antheridia and oogonia are developed on the same filament. That means the filaments are bisexual. Whereas in case of dioecious species, the male and female gametangia or antheridia and oogonia are developed on the separate filaments. Whereas in case of nonandria, the antheridia are produced on the small filaments called nonandria or male filaments, which remain epiphytically attached with the oogonium or suffultatory cells, which is present just below the oogonium. Thus, in nonandrous species, there is dimorphism in case of oedogonium thalli. That is, male and female oedogonium filaments are different in shape, and they are different also in their physiology. Now, dear students, we will discuss the development of antheridia and oogonia in case of oedogonium. The antheridia or the male reproductive organ is produced by repeated cell division of any of the cap cells of the filament. Usually the cap cells divide vegetatively or divide by usual mitotic division into so many cells transversely. Each segment or each cell divides transversely or vertically into two sperms. The sperms, after their formation, are just like zoospores in their morphology, but they are smaller in size and having similarly the crown of flagella at the base of the beak-like structure. The sperms are liberated simply by the transverse division or by the transverse rupture of the antheridium, similarly as the zoospores are liberated. Now these sperms swim freely in the water for about an hour and then finally reaches up to the oogonium. Oogonia are highly differentiated female gametangia. The oogonia develops from the actively dividing or actively growing cap cell, and this growing cap cell acts as oogonial mother cell. This oogonial mother cell divides vegetatively or mitotically into two cells. The upper daughter cell is having more cytoplasm and larger nucleus, acts as oogonium, and the lower cell acts as suffultatory cell or supporting cell. The cytoplasm of the oogonium stores reserved food material and a large amount of cytoplasm and develops into oospore or egg. In the macandrous species, the male and female gametangia are developing on the same filament. To ensure cross-fertilization, the male gametes or antheridia are getting matured just one day after the oogonia to ensure cross-fertilization. Whereas In Macrandus dioecious species, the male and female gametangia are born on two different flaps. In Nanandrous species, the sexual reproduction is somewhat specific and complicated. Here, the male gametes or antheridia are produced on small dwarf filaments called nanandria. These nanandria are actually produced by the special type of spore called androspores. These androspores are produced by any of the vegetative cells of the oedogonium, but the vegetative cell which develops the androspores usually possesses one or more caps at their anterior end. These cap cells divide transversely into so many cells, and each cell develops a single androspore. The androspores are identical like zoospores, having same crown of flagella at the base of the beak, and they are produced liberated in the same manner as the zoospores are liberated. When these androspores are liberated by the rupture of the androsporangium, they move freely above in water medium, and finally they reach the female filament of the oedogonium thallus. In Nanandrous species, after attaching themselves with the egg or oogonium or subalterine cell of the oedogonium, they germinate. After germination, they attach themselves with the oogonium by the rhizoidal cell or they hold fast, and the remaining portion gets divided by transverse division into so many cells. At the apex of this small nanandrium, certain cells develop antheridia, and each antheridium divides transversely or vertically into two sperms. These sperms are liberated from these antheridia. The androspores are produced from the androsporangia and are just like zoospores, having a crown of flagella at the base of their beak. These androspores are produced from the main filament by the repeated transverse division of the oedogonium filament. After their release from the filament, they move freely in the water and remain in search of the female filament. On the attachment with the female filament, they germinate in attachment with the oogonium or any of the supporting cell. The germling attaches itself with the oogonium or supporting cell by means of a attaching cell, or we can say it is a hold fast. The remaining portion of this androspore germling divides transversely into a small fragment, into a small filament called nanandrium. At the apical end of this nanandrium, certain cells divide transversely and form antheridia. And each antheridium produces two sperms. The sperms on maturity are liberated from these antheridia, and they swim freely in water, remains in search of the oogonium.